me, I got involved in genetically modified foods in a very interesting way 2 years ago on the front page of MIT Technology Journal, which is the most eminent technology journal, which I appeared on the front page many years ago for another company. I did EchoMail. MIT's Technology Journal in the January 2014 issue had a big front page article which said "Buy fresh, buy GMOs." And this concerned me. Why was MIT actually advertising GMOs declaratively saying it was safe? So what we did was we used cytosol. I mean, we did this really as a corporate social responsibility project. We used cytosol to literally, and we looked at really understanding the claim that Monsanto makes today is two claims. One is that the genetically modified version of what they create is no different than the non-genetically modified version. That means if they create genetically modified corn, they're saying it's materially the same as a non-genetically modified corn.
Something I could never believe.
Right. If they say they created genetically modified soy, it's the same as a non-genetically modified soy. In fact, 94% of the soy in the United States is genetically modified. The other claim they make is that when you eat this, it's not going to hurt your body. So those are their two pillars of their claims. The first one, called material difference or substantial equivalence, that the non-GMO is the same as the GMO, is used as the basis in the United States in a self-regulated way for allowing that GMO crop to be given to the public. So right now, if Monsanto, when they created the Roundup Ready soy, the GMO soy, all they needed to do was to show that the GMO soy was not materially different to the non-GMO soy. Well, how do they do that?
Yeah, that was my next question. How did they do that?
Right, so they choose criteria. So it's regulated by the manufacturer. So if Monsanto says we're going to check the nutritional value, carbohydrates, fat content, protein, the color, for example, and if they compare those with the non-GMO and they show they're the same, then plus or minus 20%, then they simply let the FDA know they've done this material difference testing. And the FDA actually has no say in allowing or not allowing GMOs. They just do what's called a quote-unquote safety consultation, and they give them a letter. And the letter says, "Dear Monsanto," and I'm sort of paraphrasing the template letter that they get, "thank you for doing the testing. You state you have done the testing," you meaning Monsanto. "You state that there are no material difference and that really the responsibility is on you. But because you say you've done the testing, we're fine with it being allowed to go to the public." That's it.
And we're talking millions of people eating it on a daily basis.
Right. So the point is that the issue is, how did they do that safety testing? E.g., how do they determine the material difference is based on criteria that they choose. Okay? So that's the point here that everyone needs to understand. So this is no different than if you went to the Department of Motor Vehicles and you're trying to get your inspection sticker and you told them, let's say for emissions, "Hey, I did the emission testing in my home and I find out based on the criteria that I've set that the emission's okay." And the Department of Motor Vehicles says, "Thank you, Shiva, thank you, Elaine, for doing the testing. You can take the car on the road," because their whole mode of allowing the car on the road is just based on you saying you did it. Okay? This is probably the most important point everyone needs to. Mainstream media hasn't gotten this right. In fact, the anti-GMO activists haven't gotten this right, neither of the pro-GMO. Everyone's confused this very fundamental issue. There is no standards for safety testing of GMOs. It is all based on self-reporting. I'll repeat that. There's no standards for safety testing of GMOs, all based on self-reporting. In fact, the GMO salmon, if everyone reads, it says that the company determined that it was nutritionally equivalent. The company determined it. how do they determine it? They probably establish their own criteria. Well, the fat content, the protein content was the same, therefore it's safe. Okay? So what we did, we actually did science. We actually went and did science. And what did we do? Well, every scientific organism, your body, a plant, a bacteria, any animal, is literally a walking, breathing chemical factory. Right now, you have millions of proteins and molecules in your body that are interacting, that define you. A woman, for example, is different than a man based on the fact that a woman has certain chromosomes, XY chromosome for a man, a woman has XX. So if you go down to the molecular level, you can actually establish objective criteria. What makes, let's say, a woman different than a man? Okay? What makes an ant different than a cockroach? There are molecular fundamental reasons. However, if you randomly said that we're going to use a criteria of does a woman and a man have hair? Do they both have two eyes? And do they both have a nose to determine gender? You'd say a male is equivalent to a female, right? It would be absurd because you're not using any science or what are called objective criteria. So when we did this research, so that's the background. So we wanted to find, can you establish criteria for really understanding the difference between a GMO and a non-GMO? Like scientific criteria, not stuff that you make up so you can convince people that the GMO is the same as a non-GMO. Like, what are the criteria?
Well, and with the technology we have today, you would think that should be possible.
Right, but the issue is you have to do a systems analysis, okay?
And that's what you specialize in.
That's what we do.
Thinking about systems so that we're actually getting it down to a precise measurement.
Well,
scientifically.
Systems thinking means you look at the whole, okay?
Okay.
Systems thinking says you got to look at the interconnections. How are things interconnected? Because when you look at not the parts but the interconnections, then you can understand the whole. Okay, that's systems thinking.
And one reaction is influencing another reaction as well, and there can be a cascade of reactions that happen when you change even one small thing. And I think that was something you said in your paper, which really hit home with me. And that's what I believed all along, and that's why we've had this show all this time trying to get that point across. I'm so grateful to have found someone that can articulate all of this in the way that you do, because I think we just need to get what you are trying to explain to us across, and we need to just keep saying it over and over again and explaining this to everybody else out there in this format. So why don't you go ahead and
Right. So what we did was we wrote a series of 4 scientific papers which were peer-reviewed and they were published. What we wanted to understand is what is the difference between a non-GMO and a GMO, and how would you establish that difference scientifically? Okay. So in paper 1, we went through 11,200 plus papers to find out, are there certain molecular mechanisms common to all plants? And we found out there's a metabolic mechanism called capital C1 metabolism. What is that? To put it simply, it's how plants create carbon, right? Photosynthesis occurs and plants create carbon. C1 metabolism, think about it as a big engine that the plant has. That engine is composed of 3 major parts. One part is called methionine biosynthesis. You don't need to understand the details. Methylation, and another part is called formaldehyde detoxification. So you can think of them as 3 different gears which are working together for this thing called C1 metabolism. Within each of those gears, there's even more molecular systems. So for example, the methylation process creates formaldehyde. Okay, so all plants actually are creating formaldehyde, which is a known carcinogen. However, plants are very smart. They have a formaldehyde detoxification sub-engine, which takes the formaldehyde and detoxifies it using a chemical called glutathione. If you look up glutathione, it's one of the most important antioxidants that occur in nature. So plants do create formaldehyde, and then they detoxify it. Okay, so we found in those 11,000 papers, we were able to connect these papers when we found the molecular mechanisms, all the different gears involved in C1 metabolism. That we published in a journal called Agricultural Sciences. In fact, my research team did it, but we also did a very cool thing. We worked with 4 high school students and their teacher in my old alma mater high school, Livingston High School in Livingston, New Jersey. And these students were the best of the best AP chemistry students. And by the way, Livingston High School is one of the, I think, top-rated public school systems in the entire country. So that was paper 1. Then in paper 2, we took all those molecular mechanisms, which are the gears, and we used cytosol to connect all this research together. Now remember, all of these molecular pathways are not something we made up. They're not a model. They're actually from many, in fact, 6,497 actual lab experiments that scientists have done across 184 institutions in 23 countries. So with this technology, we're able to take all this research, imagine them being 6,497 blind men, and connect them together, okay? So now when we connected all of this together, what we find is that in normal plants, we're able to show this, that formaldehyde is created and it's detoxified. And glutathione, that antioxidant, is beautifully maintained in normal plants. So basically, when you look at any time, if you pick a plant, it's always going to have some amount of background formaldehyde because some of it didn't get detoxified, okay? But the point is,
and isn't that like a waste product? It's just like a byproduct.
It's a waste product. It's a waste product, yeah. Okay? So the point is, when our final paper came out, some of these detractors who were trying to disinform people and confuse people, they said, oh, all plants have formaldehyde. Yeah, they do, but that's a background level, okay? But then in the normal case, plants create formaldehyde and they detoxify it. So that was paper number 2. We published in the American Journal of Plant Sciences, peer-reviewed journal. In paper number 3, we added another capability. So we had the formaldehyde detoxification pathway, part of C1 metabolism. Then what we did was we found out that plants also undergo stress. Just like you get stressed out if the weather changes or you have emotional stress or you stop eating. Well, plants undergo stress, for example, when the weather changes or when there's a drought. If there's a drought, a plant undergoes what's called oxidative stress. It thinks it's going to die. So what does it do? It's no longer in the normal mode. It goes into the oxidative stress mode. And in that case, the plant will use up its glutathione, which is that antioxidant because it's under oxidation. Glutathione levels will drop, and guess what happens? Formaldehyde will accumulate. Again, nothing to do with GMOs. This is a natural phenomenon that occurs when a plant is stressed out. That was paper number 3, which we also published in the American Journal of Plant Sciences. So we did paper 1, 2, and 3. Then in paper 4, which is, I believe, published in July, we asked an important question. We said, does genetic modification, and we focused it on soy, does genetic modification disturb the natural process of plants? And we felt systems biology could help. Remember, we'd done these 3 papers. So what we did was we said, we decided to take soy to be very specific. Remember, genetically modified soy is a number one genetically modified organism in the United States. 94% of all the soy we eat is genetically modified. So what we found was, again, we didn't make any of this up. We found another series of many papers where people had conducted laboratory experiments when they modified soy, did the genetic modification, what was called Roundup Ready soy. Remember, this is a Monsanto version where they create a version of soy which can resist their herbicide called glyphosate. So therefore, you need not only their pesticide, but you also need, they own both sides of the equation here. You also need their soybean. So Roundup Ready soy, which is a modified soybean, we found research when that soybean was modified that 5 different molecules were disturbed. They were not the same as a non-genetically modified soy. There were 4 enzymes. There were 4 enzymes and hydrogen peroxide. Okay, so this is independent of us. Independent research has shown 4 enzymes, proteins, get modified, disturbed in the genetically modified soy, Some of them are higher, some of them lower than the non-genetically modified. So we took that result and we integrated it with the results of paper 1, 2, and 3. And what do we see? What we see, again, it's all integration. What we see is, we see that the plant believes it's under stress. So it uses up its glutathione, and guess what happens? Formaldehyde accumulates. So the same process when the plant thinks it's in a drought mode is what that genetic modification is showing. So remember, again, we had integrated 6,497 wet lab experiments. We're not making this up. Across 184 institutions in 23 countries. From that integration, we see this dramatic growth, accumulation of formaldehyde. It's no longer detoxifying it, and this drop in glutathione. Well, in the fourth paper, we said, look, we're noticing this, and we're saying, and we concluded by saying, yes, we need to do more research to validate our findings. So we're very honest about it. And we furthermore said that should not glutathione and formaldehyde be used as objective criteria for determining the difference between a genetically modified food and a non-GMO? All right? So what we're saying is, forget nutritional content, fat, carbohydrate, blah, blah, blah. Why don't we use, maybe there are others, but definitely our research are saying formaldehyde was very different in a GMO and a non-GMO, and glutathione was different. But Monsanto or others are not using them as criteria, when in fact they should be, because they're such important molecules. And when we concluded our paper, we said, look, if we can't, if we wanted to grow soybeans, the original non-GMO soybean and the GMO soybean, and we did the testing, and in fact we validated our predictions, we would be attacked, because people would say, oh, what dirt did you use? You didn't use the right seed. Obviously, companies like Monsanto would likely attack us. And the reason they're able to attack us and protect themselves is there are no standards, right? So because if you have no rules, it's a Wild West. And the FDA has set it up so it is a Wild West. There are no standards, so anything goes. So what we have said was, to bound that problem, you must have standards. So what we're saying is we must have standards, which means if you're going to test a non-GMO GMO, because remember, material differences used as a basis of safety, that we need to have scientific standards. What's the soil you should use? Where should the seed come from? How long should the seed grow? How should you do the testing? So whether I do that testing, or Monsanto does that testing, or MIT does that testing, or Harvard does that testing, or any lab does the testing, or even high school students in Livingston, New Jersey, the results should always be the same, because you're following the same protocols. Because those protocols don't exist, things are being put out without any real testing.